Hello guys, welcome to the Inspiring Minds. Now we are going to watch the second video of microbiology. In the previous video I told you about the gram stain. First of all, I would like to make it clear that the staining process is not only done for the bacteria. Okay, it is applicable almost all kind of microorganisms, but their names and chemical properties and structural properties are different. Okay, so our today topic is morphology and physiology of the bacteria. See, why bacteria is so important? Bacteria is that microorganisms which cause most of the disease, infectious disease, which is very harmful for the human being or any other animals. That's why we study. Okay, so I will take it from those link from that link from where you lost that concepts. Not lost, if I tell you that practically that was the time when you studied about the Monera, kingdom Monera. Okay, unicellular prokaryotes. Unicellular means one cell, okay, and prokaryotes, as you know, they have false nucleus. False nucleus doesn't mean nucleus absent, okay, or nucleus absent but nuclear material present. False nucleus means when we say that there is something false appearance, false chemical test, false diagnosis approach. That means that there is something like that, but that is not exactly that what we supposed to see. Okay, so as the Monera kingdom, known as the unicellular prokaryotes, if I tell you their peculiarities, these were they absent the membrane bound organelles, that are the basically endoplasmic reticulum, mitochondria, lysosome, etc. Okay, so and the second one, they have 70s ribosome, and third one, they have 0.2 to 2 micrometer diameter. They are that much small. See, they are in micro level. That's why we can't see them our open eyes, naked eyes. Okay, so at this moment, that's why we are studying microbiology. So here the prokaryotes contain false nucleus. That's why that is known as nucleoid. They contain nucleoid. You can't again I want to focus here that you can't say that they don't have nuclear material. They have nuclear material, right? So the first point, there is lack of the membrane bound organelles. Second, they have 70s ribosome, and third, they have a diameter of 0.2 to 2 micrometer, and there is single loop of DNA. What does it mean? I want to tell you that these things which you can found in other books, I mean almost all books when you study the microbiology, but here are the things where I'm going to explain you what are those things which we study there. 70s ribosomes, actually it is the structural unit, how we found that ribosomes inside cell. And single loop means when we talk about loop, simply loop means like this way. Okay, loop of DNA. If you break the DNA, deoxyribonucleic acid, they are double stranded. So simply means they are going to be like this way. Okay, it is a circular loop. Here it says single loop. Single loop means there is no any point for start and end. It means that it may be like this way, or it may be like in cluster form but in loop form. Okay, that means single loop. And the third one, there is extra chromosomal material present in the prokaryotes that is known to the plasmid. I will tell about plasmid in my further lecture, in my further video, because it is very important and very infectious for the organisms to cause the disease, and in the pathogenesis study it's very important. As similarly we have, eukaryotic cells, they have mitochondria, extra chromosomal DNA material present. Then it is a formation, means that how the new bacteria, that new prokaryotes form. one of the most important thing, and it is my personal observation that many times we are confused with that things, that we simply say that there is no, there is asexual reproduction, or simply we stick with the one word, there is only binary fission. So make it clear that yes, when someone asks you that can bacteria have, can any bacteria have sexual reproduction, means formation of gamete and transfer of genetic materials, so you can't say that no, all bacteria are asexual. No. Thing is that there is always an exception when you study the science. Okay, so keep in mind that mostly they transfer their genetic material through the process that is binary fission. As I think you all know that binary fission means that when one cell divide and further divide, that is binary fission. Okay, but the transfer of genetic material is also it is much more similar and almost similar to the sexual reproduction. When plasmid transfer the genetic material, then their exchange of genetic material, then there we can say somehow similar to the sexual reproduction. So it will be wrong if you say that there is only and only strict to the word that asexual reproduction. Okay, so if we talk about prokaryotes further divide into archaeobacteria and eubacteria. But if you go there, the proper classification and physiological properties, so you can say that archaeobacteria are capable to survive in the extreme temperature. When you study further eubacteria, the true bacteria, then what you will find? They form the sporulation process, and at that moment, what they do, they are also able to survive in extreme high temperature. So existence is possible. That doesn't mean in high temperature only and only archaeobacteria found. Spores of eubacteria can also be found. Okay, so that is the second point where you should be clear that only if you see the temperature, and there is in MCQ questions that if you see the archaeobacteria and eubacteria, then definitely you have to go to the archaeobacteria. But if you found in multiple options there is also spore, then this one may be probably the second most priority, because the spore are highly resistant to the temperature and also to the pH. Okay, so let's go to the eubacteria shape, because we are interested, and also it's in our syllabus that we have to study about eubacteria. Eubacteria known as true bacteria because there is existence of nucleate shape. See, I tell you honestly that if you are studying in a medical college, never expect things to be simple in front of you. When you study these things in your high school and intermediate, that was that much simple I kept in front of you. But if you go to the staining process and the laboratory process, then definitely it's going to be much more drastic change. Okay, so here I am telling you just a rough diagram. Later I will go to their cultivations, as in my previous video I told you about gram stain. That is only one stain method. There is stepwise and a lots of staining method. They have first I will tell you their shape, that as here bacteria have 0.2 to 2 micrometer diameter. It's simply, I think you all know diameter, and when we talk about the length, that may exceed to 5 to 10 micrometer. Okay, they have outer structure that is known as flagella, also known as flagellum. Okay, and they have a small cellular extension from their surface that is known as pili, and the outer one there is capsule, also known as capsid. Somehow we call it cap, and after capsule there is cell wall, and after cell wall there is plasma membrane that we known as nuclear membrane. Yep, plasma membrane. Okay, these are the simple structural component of a bacterial cell. Okay, once again I am going to make it clear. Unicellular and prokaryotes proper nucleus absent. We are talking only about them. Next, there will be the existence, their properties, and their further, chemical and structural properties. I will tell you stepwise. As I told you in my previous video that there is gram positive and gram negative bacteria. These are the only categories of bacteria which we can decide on the behalf of they having this kind of thick cell wall and cell wall property, and they have those particular properties and other they don't have. That's why they are gram negative bacteria. Okay, further class